Hi. In this series, we're talking with the scientists from the Dangoor Center for Personalized Medicine to get an up-close look at what happens in their research labs, what developments we can expect to reach the market, and what medicine might look like in the years to come. This time, we welcome Professor Mira Barda-Saad with us. Professor Saad, you did much of your training at Bar Ilan. You got a degree in life sciences as an undergraduate, graduated with honors, and then a master's in microbiology, again with honors you graduated, and then your doctorate even. You studied there in conjunction with the Weizmann Institute, and from what I understand, you graduated with high honors. Then you continued on to a postdoctorate at Weizmann, as well as one at the NIH, the National Institutes of Health in the United States. So welcome. We'd love to talk to you today a little bit about what's going on in your lab. I know that you're an expert in immunology and immunotherapy, in particular as it relates to cancer. So perhaps we can start with some basic terminology that you could lay out and explain to all of us. What do we mean by immunology, immune therapy, cancer? There's some terms that are thrown around like natural killer cells. Maybe lay out some of the basics for our conversation.
So thank you for introducing me. So we are actually focusing on cancer immunity, and specifically we are focusing on the immune system, trying to develop a novel immunotherapeutic approach to combat cancer. So immunotherapy is actually technology in which we empower the immune system to better fight cancer. So we are using our own immune system and boost it in order that it will fight cancer much more efficiently with much less side effect. And I would like to give some terminology in order that our conversation will be much more fluent. So there are several cells that play a role here in this game. The first one is the natural killer cells. Mm-hmm. The natural killer cells are first line of immune defense against cancer and also viral infection. The big advantage of natural killer cells that we are focusing on NK-based immunotherapy is because that these cells we do not have to sensitize them. We do not have to introduce the pathogen to these cells. They know how to recognize it and to release their weapon towards this pathogen. So this is a huge advantage over other technology like T-cell lymphocyte, T-cell-based immunotherapy, that in this approach, T-cells should be sensitized to the pathogen or antigen part of the pathogen in order to work. So natural killer cells, they are part of the innate immune response, in contrary to T-cells that they are adaptive immune cells, and they actually have some motifs, some receptors over their surface that recognize some motif of pathogen. And once they actually screen the body and they find that there is invasion of a pathogen into the body, meaning cancerous cells or are being developed, are being transferred, or a virus, they actually start to behave like there is a foreign attack, and we should all fight this and mitigate it. And as I mentioned, other cells are T cells, either helper T cells that they produce some factors, some cytokines that help the immune cells to be much more responsive, and some other cells are B cells that actually eventually secrete antibodies, are part of the humoral immune response that help the antibody help to actually phagocyte or to swallow the pathogen and to remove it from the cell body, make it much more easier for the immune system. So all these units actually work very nicely together. They are complementary angles.
So when we talk about immunology, we're talking about all of these different possibilities. But from what I understood from you, the natural killer cells are in some ways best naturally suited on their own to fight against any pathogens. And in pathogens, you included both cancer, since they're cancer cells, since they're not supposed to be happening. Pathogens essentially are things in our body that aren't supposed to be there, or viruses as well, correct?
Exactly.
And as an immunologist, you're trying to strengthen some of our natural responses that our body automatically produces, but make them better at doing their job.
Exactly.
So what exactly is out there right now? I know in 2011, the FDA, the Federal Drug Administration from the United States, approved a number of different immunological treatments for cancer. What exists on the market today?
So there are actually, I would say, three major immunotherapy approach. First one is inhibitory checkpoint, which I will expand much more about it because our strategy belongs to this category.
Okay.
Second one is adaptive cell transfer, and third one is cancer vaccination. So for the first one, inhibitory checkpoint, so we actually realized during the years that the tumor microenvironment actually secretes some factors that inhibit the immune system. So once we realized that, it was much more easier to develop a immunotherapeutic approach to fight this. Cancerous cells are very tricky. I mean, they have some tricks in order to try at least overcome the immune system. One of the tricks, they try to downregulate molecules that the immune system recognizes in order to fight them. So they either lose them or mess them. Another strategy is to secrete, as I mentioned, factors that associate or bind to the immune cells and inhibit their activity. And third is actually they also sometimes it's not just factor, it's also ligands that associate with the receptor of the immune cells. So there are several tricks, several tools that they use in order to downregulate our immune system.
That's fascinating, because I always just thought of cancer as something that grows. They're extra cells. But what you're saying, and I won't attribute consciousness to it, although as a layperson, it almost feels like they're these nasty invaders that are trying to fight and take control of something deliberately. And obviously, it's a biological process. It's not but it is aggressive. They're not just growing. They're trying to make sure that our natural responses to fight against them through our immune system are suppressed.
Exactly.
So that we have to not just combat the fact that there's this pathogen that's growing, but we have to fight against what they're doing against our immune system.
Exactly. It's an army, actually. I see it as a fight between armies, between our immune system to the pathogens, to the cancerous cells, and it's a real fight.
And they're fighting back.
Yeah.
That's fascinating.
So I think we should gain much more knowledge. Once we have more knowledge on the cancerous cells, we can actually come up with new strategies that will overcome their tricks.
Do you focus on, there are lots of different cancers, do you focus on a particular cancer in your laboratory?
We do work on specific cancers, but let's say that our wish is that our strategy will be cross cancers, cross patients. This is, I think, the most top goal of all cancer biologists. Obviously, it's a very ambitious goal, but definitely this is our goal.
But do all cancer cells act in a similar way so that you can learn how to fight against them as a group?
So that's another huge advantage of natural killer cells over T cells, is that their fight against cancer is actually quite similar. They actually recognize that we're talking on, once they, as I mentioned, screen the body for pathogen, and they found that cancerous cells is interacting with them, they actually release their weapons. The weapon is like We call it cytolytic granules. It's actually like vesicles, contain enzymes and other materials that penetrate the cancerous cells and induce their suicide.
Wow.
Meaning they actually tell them you should die in a very short time. And actually within minutes, the cancerous cells start to die. So we can, in my lab, follow this interaction, following this crosstalk between the natural killer cells to the cancerous cells. Obviously, we are familiar with the molecular mechanisms that are being involved and trying to enhance this reaction of the natural killer cells towards the cancerous cells. So now that we realize that the natural killer cells have not just activating receptor, but also inhibitory receptor that the cancerous cells use in order to downregulate their activity, what we are doing, we're suppressing the inhibition, suppressing the suppressors in order to get activation.
So this is fascinating. It truly is, because I mean, I'm interested in science, but even for people who aren't fascinated by it, the language you're using, it's two armies confronting each other, trying to suppress each other. Then there are these killer cells that are inducing suicide. It's really, the way you speak about it is it really elevates it outside of, working a little petri dish here, which is how most of us grew up studying microbiology. How close is your lab to coming to something that can be brought to market?
So actually, in the last few years, we focus on 3 strategies. The first strategy, we develop nanoparticles that encapsulate materials that can empower the natural killer cells. So we do not have to isolate the natural killer cells from the patient body, like the common strategies. We just, these days, inject the nanoparticles to the tail of the mice. They actually recognize the natural killer cells, integrate into them, and manipulate their cell behavior to become much more vicious. So we are taking like Nebuch cells and transform them into serial killers, that they could better fight this cancer. And this is one strategy, which obviously involves nanobiology. Another strategy is that we characterize the natural killer cells, and we found that actually major part of them are actually dysfunctional. Major part, once the natural killer cells reach the tumor site, sometimes they do a lot of efforts to kill the cancerous cells, and as a consequence, they actually become dysfunctional. They become exhausted, they do not do nothing. So we actually characterize this population, and via these nanoparticles, we actually encapsulate the nanoparticles with some materials that can rewire these natural killer cells and transform them from dysfunctional cells into functional cells. We know which molecular mechanisms are involved in inducing exhaustion or inducing dysfunction, and these we actually target these key molecular cascades and transforming them to be much more active at the tumor site. So this is another strategy. Third strategy actually associate more to blood cancer. And there we're using AI, artificial intelligence, and machine learning algorithm. We actually identified small molecule compound, drug-like based small molecule compound that can actually cause downregulation of a protein which is part of the cytoskeleton of blood cells, of hematopoietic cells. And once we goes to degradation of this protein, the cells cannot become activated, they cannot proliferate, they cannot migrate, they cannot invade the tissues. This is huge because this protein that we target via this small molecule compound is actually exclusively expressed in hematopoietic cells the small molecule compound recognize just the active conformation of this protein. The protein is called WASP, Wiskott-Aldrich syndrome protein, and it's mainly in cancerous cells. So we do not have any off-target towards naive cells that are not involved or normal cells. It's just target the cancerous cells. And this is definitely a real breakthrough. So now we are actually in the stage that we almost finished the preclinical phase. And obviously there is a very lengthy way ahead of us until it will be a drug that is suitable to treat the patient. As we know, we have to check it both from safety and efficacy in patient, and this take years.
So you have these three different things. The first two are working with strengthening the natural killer cells, and the third one, if I understood correctly, wasn't about natural killer cells. It was about targeting a specific protein that's associated with a blood cancer. When you talk about trials, are they three separate trials? Are you combining them together to be most effective? How is this working?
So it's actually three different strategies. Strategy deal with different immune cells. And so we cannot combine trials, actually. We have to treat them three different approaches. And obviously, each of them come from a different angle. I mean, the first one is developing tools, delivery system of the drug. The second one is dealing with cells which are laid back. By the way, this strategy, the second strategy, was published 2 weeks ago in EMBO journal. And thank you very much, and was chosen to be the cover of the journal due to the novelty of the technology and novelty of the data.
This is the one where you said we take the nebbish, the pathetic cells, and we turn them into warriors.
Yeah. And the third technology is actually dealing, as I mentioned, it's more exclusive to cancerous, to blood cancers, and we are speaking on small molecule compound. By the way, I don't know if we are aware to this, but most of the drugs for cancer immunotherapy are small molecule compounds. I mean, part of them at least. So they are very effective because of their low molecular weight, and so they can actually integrate into the cell very easily. And obviously, I would say that there are two motives that we do pay attention to them, that it should be first highly specific, and secondly cause much less side effects.
Right. That's the personalized piece of this.
Exactly. In contrary to the common or conventional approach like chemotherapy,
you just sort of throw
radiotherapy, that it's definitely effective because it's killed the highly proliferative cells, but the side effects are kind of huge.
Yeah, all of this is very personalized. It's very much if it's the natural killer cells who are targeting just the pathogen, or if you're introducing this small molecular compound that's targeting the protein that's associated with the specific cancer.
Yeah, the protein in a specific conformational structure. I mean, not every type of the protein. I mean, only when the protein is highly activated and it's an open conformational structure, it goes to its degradation. When the protein is closed, like it is in naive cells, it does not interact with this.
So it's only the cancerous cells in the state of cancer that it targets.
That's what we've proven, and that was published like 2.5 years ago in Nature Communication as a novel strategy.
Amazing. Now, a lot of us heard about immunotherapy in 2020 with the mRNA vaccine for corona. Is there any connection here with what you're doing and what that vaccine was? You know, there was a lot of talk around the science of it, but I think it's still a little fuzzy. So if there is some way you could explain it to us, and if it's tied to the work you're doing, that would be great.
So the delivery system is not identical, definitely, but similar. I mean, liposomes, which are lipid vesicles, which encapsulate, as I mentioned, our materials or our factor in order to empower the natural killer cells. So with the vaccination that we got, actually, it was encapsulated in vesicles that were injected to us in order that the immune response will be empowered towards the virus. So there are some similarities, but also some differences. And it's very important to mention that cancer are heterogeneous, meaning they are very heterogeneous, and we have to...
What does heterogeneous mean?
Heterogeneous, they are so different one from another. One type from another, and we have to deal with this. So this is definitely an issue that we should take into consideration, as we mentioned also at the beginning of our talk.
Yes, although the natural killer cells will identify all cancers, you're saying.
They have this potential. They have this potential because they actually have like a general mechanism that secrete these cytolytic granules or operate their killing mechanism.
That fights against all pathogens, not just a particular one.
That fight against types of cancerous cells. By the way, these days we know that the natural killers, there are some strategies of natural killer cells, and we see that they are much more effective towards hematologic malignancies and much less towards solid cancer. Mm-hmm. And that's because of the fact that solid cancer, they have some difficulties to penetrate into it. So there is a huge difference between stage 1 cancer, solid cancer, to stage 4 solid cancer. I mean, the more the cancer is much more stiff and bigger, the natural killer cells find some difficulties to penetrate into it and to release their weapons. Sometimes even they become exhausted, as we mentioned earlier. So we have to overcome this. We have first to expedite and to give them much more strength to penetrate into the solid cancer, and moreover, as I mentioned, to rewire them from exhausted cells to functional cells.
When you say solid cancer, you mean tumors?
Yeah.
Okay, as opposed to like blood cancer.
Exactly. Okay. We're working a lot on pancreatic cancer, on lung cancer, and others.
Understood. So there are different kinds, different treatments, but you're working on a number of treatments. So hopefully, as you said originally, hopefully you'll be able to address all the cancers at some point.
That's the goal. But as I mentioned, it's very challenging goal, and but we are trying to do our best.
You always have to aim high, and then even it sounds like even the incremental steps are so innovative and critical that if you and your lab don't get to the final goal of resolving the issues of all cancers, along the way you will have done incredible things. So maybe we can talk a little bit about you. Now that you've been very serious and professional and told us all about your research, tell me a little bit about you. Did you always want to be a researcher?
I think I did. I mean, since I was really a child, I think less than 10 years old, I read a lot of books, and I try to expand my knowledge specifically in science. I remember myself really very much interested in almost everything that relates to science. I'm a very curious person, and curiosity drives me. By the way, I can read on insects and still be fascinating, try to find the mechanism that work them, drive them, etc. So I never studied, as my son Daniel said the other day, Mom, you never stop studying. I mean, I'm all the time trying to expand my knowledge. I think knowledge is really very powerful in order to come up with new technology, come up with new strategies. You must be very much aware to what is going on these days. Not a month ago, but yesterday.
And also, I know a lot of innovative breakthroughs in science come from things outside. So reading about insects, maybe there's some incredible mechanism within the insect that you can apply in your lab towards fighting cancer.
I mean, there are some things, some motifs that you can definitely adapt from other systems and make your system much more powerful. Definitely.
That's fascinating. So, okay, you always studied nature, you were always interested in science. How did you land, as somebody who's curious about insects and all of nature, how did you land on immunotherapy and cancer?
So, unfortunately, very close people to me were diagnosed with blood cancer. And I was an immunologist from, the time that I remember myself. But I mainly focused on basic science. When I was at the NIH, at the National Institute of Health, I mainly worked on basic science. So when I came to Israel, I decided that I should do also much more applicative approaches and not just basic science, although I must say that at the NIH, although it's like one of the largest medical centers in the world with innovative approaches, they do put a major focus on basic science because they know that this is very important in order to find new strategies. So I did a little bit, I shift to applicative approaches, and that's what we are heavily doing now, or in the last, I would say, 15 years.
And it's bringing about some pretty extraordinary advances. It makes a lot of sense, and we're all very grateful for the work that you're doing. Where do you see science broadly, or you can be as specific as you want, the fields of immunology, cancer, where do you see it in 5, 10 years?
I think I touched it during my old talk because I all the time think on the next immunotherapeutic approach. I all the time think about it. I mean, this definitely what drives me. So as I mentioned, it should be highly specific, much less side effect, much less toxicity, a treatment that will actually expand or the survival of the patient and definitely improve their quality of life. That's the way I think all of us see it. And in order to do that, our tools should be much more elegant and much more, target specific.
You left the NIH to come back to Israel and Bar Ilan. Do you feel like the field, your field, is really advanced and forward pushing in Israel as much as in other places, and at Bar Ilan in particular? What brought you back? I know you're sort of a child of Bar Ilan, but what brought you back in the end to Israel and to Bar Ilan?
So Israel is my place. Israel is my country. I think I should state it very clear, definitely in these days. I think Bar Ilan University, on one hand, give us the environment which is highly professional to do good research, on one hand, and on the other hand, also it's a very friendly environment that encourage us to do cooperation and to have really good discussion.
That's wonderful. Well, Israel's lucky to have you. Bar Ilan is lucky to have you. And I hope you continue to move forward with the amazing work you're doing that will help millions of people around the world. Thank you so much for joining us today. It was really a pleasure to speak with you.
Thank you for inviting me.